Wonderful. Well, everybody, welcome to another Precisionary Webinar. Today, we really have the honor of having Dr. Kleanthis Fytianos, and I'm so sorry I mispronounced your name, but he is going to talk about a very popular topic that a lot of our clients at Precisionary have asked us about for precision cut lung slices, or PCLS. So he's going to talk about heme-induced injury in PCLS. A little bit about Dr. Fytianos. He completed his PhD in biology at the University of Fribourg in Switzerland. And he actually is joining us with a big time difference. So thank you again.
No
problem. During his PhD, he investigated nanoparticle interactions with human immune cells and also made advancements in vitro models for respiratory disease. Afterwards, he pursued a postdoc research period in biomedicine and translational medicine with a focus on respiratory disease models, both in vitro and in vivo. And since 2019, he has worked for CSL Behring, the Research and Development Department, as a senior scientist. He currently leads several projects in the respiratory therapeutic area that are at preclinical stages. And this is particularly relevant given that we had the COVID pandemic recently. His team focuses on relevant and physiological in vitro and ex vivo experimental disease models that could support later stages of development. And we are really excited to have him today to talk about the heme-induced injury of precision cut lung slices. So with that, Dr. Fytianos, I want to hand it to you. And for all of our attendees, please feel free to use our chat box to make comments or ask questions. You can do it both publicly or privately. And after Dr. Fytianos' webinar, we'll have a Q&A session, which I'll moderate. So you can show yourself and ask questions directly or enter them into our chat box, and I'll be happy to read it for you if you're a little bit shy. With that, I want to give it to Dr. Fytianos and say thank you again.
Thank you for having me. It's very nice to be here with all of you. And yeah, also thanks for the nice, the comprehensive introduction. Maybe before I start, just a brief disclosure that I work for CSL Behring. I'm an employee of CSL Behring. All the data that I'm going to present today has been published before. But yeah, as you said, I'll be happy to further discuss on the Q&A session. And without further ado, let's move on. So today we're going to talk about heme injury. For those of you that you're not familiar with it, heme is a product of hemolysis. So the red blood cells, like under some certain pathologies, are ruptured and cell-free hemoglobin is released into the bloodstream. And then cell-free hemoglobin is further degraded into heme. And evidence in the literature, both in vitro and in vivo settings, has shown that it's both cytotoxic and pro-inflammatory. Taking into account some translational studies, we've seen that elevated heme levels are implicated in several conditions with hemolysis underlying pathology. Sepsis, like is a common example, and also that elevated heme levels have also been associated with higher mortality on a clinical setting. For the purpose of our experiments, we use mouse and human PCLS. So it is a 10-dimensional translational ex vivo model, and currently not much is known in terms of heme injury on a lung setting. So most of the studies, as you will see, is mainly in the liver, because most of the proteins are metabolized there, including heme. But in some cases where we have liver failure, that's why we've got excess levels of heme. And yeah, in this case, we see that not much is known on neither on a lung setting and nor on on lung specific endpoints. So this is why we want to use heme on translational material, on patient material, both human and mouse, that we will see later on. And yeah, as I said, we aim to characterize the effects of heme on ex vivo human lung setting. However, initially we started with mouse, and the reason is just to get some optimal concentrations and time points and so on, so to more accurately design our experiments for the human samples that we don't get them, you know, on demand, right? So really depends on the surgeries, and as you mentioned, like with COVID, which had like a really significant reduction of surgeries like from lung transplantation or resected tissue. So yeah, that actually could hold us a bit back. However, in this slide, I would like to show you like a high-level overview of our experimental approach. As I said, we make sure that we get access to either mouse or, more important, like human lung, and then we use the precisionary compressome, so to generate the lung slices according to the provided protocols. And then we use 24 well plates under submerged conditions with using of recommended medium, and we leave the lung slices to rest for 24 hours. After 24 hours, the excess of lung slices are frozen and used for future experiments, and also to create kind of a mini biobank, if you wish, but of course for future projects and other experiments. And we take our lung tissue from culture, and then we change the medium, and we stimulate with heme at given concentration for 24 hours. And it's really quite a plethora of assays that we could perform, both on from supernatant collection. So we use the multiplex cytokine arrays or ELISAs for more sensitivity, as well as the XTT assay for viability. While we take the tissue itself for RNA extraction, total protein quantification, and of course immunofluorescence staining. On the next slide, I would like to show to you the donor characteristics. I think it's also important for the audience, like to see what is the profile of the donors. So the last 2 years, actually, that we were working with this system, we managed to get 12 donors. You see that we're talking predominantly about male subjects. In most of the cases above 55 years of age, and all of them, or most of them, are diagnosed with oncology related indication, and as you can appreciate, also most of them are heavy smokers. Sure, now you can argue that look, it is a disease of like aging related disease, and maybe there is cell quiescence and so on. Yes, all of these are very valid points, but at least we have to start from somewhere, right? So it's important, like, to get access to human tissue. And since we're collaborating with a university hospital, it's the Department of Thoracic Surgery that is performing these operations. And usually some part of the tumor lung is removed, is resected, and the tumor-free part of the lung or part of it is dedicated to research. And this is how we get the access to it. I mentioned before about the frozen tissue. I'm sure most of you have experienced that if you use it fresh, it's always better. It gives you much better responses, but sometimes, you know, the times that we get it are not so convenient. So, for example, on a Friday afternoon, you might be notified that the tissue is available. So sometimes it's unavoidable, like to have to freeze the tissue, because either you get like a very small piece of tissue and you cannot work like with all the treatments that you may have planned before, but other times you get quite a big piece of lung. And for example, I can remember that in was one case we got more than 100 slices from one donor, and in this case, we had to freeze this tissue. And on the next slide, I jump back to the mouse, and in this case, we'd like to share some preliminary data with you. Initially, we stimulated with, as I said, we stimulated with heme at different time points. And as you can see on the viability assay, at 24 hours, we have quite a strong reduction, a significant reduction in viability. And on this data, on this time point, we see quite and upregulated levels or elevated levels of TLR4 pro-inflammatory cytokines. And on the same time, we've seen some elevation of levels of both RANTES, so this is a lung epithelial injury marker, and angiopoietin-2 is upregulated like very elevated, like significantly. And this is actually also a biomarker for sepsis, and you know, for and very important marker for endothelial injury. So we've presented these results in the European Respiratory Society annual meeting in 2022. And based on these findings, actually, this was the basis like to start testing like the human to confirm or to validate these findings in a human setting. And this is like a first glimpse of the results we got like with the human PCLS. Again, with the XTT assay in terms of viability, we see that this like from 100%, like after 24 hours of heme stimulation at 100 micromolar, is almost, the viability is almost reduced in half. Maybe just to comment on the concentration, these levels actually are corresponding or are in accordance with some real life cases. So for example, some patients that are in sepsis or septic shock have levels that this have heme levels or free heme levels at this range. However, concentrations higher than that are rather unrealistic, I would say. However, they might be useful from a toxicological point of view. If someone wants to do like a toxicology study and you need like a very high concentration, this is where you could make use of these concentrations. But if we stick more to the more physiologically relevant concentrations, again, we see quite some strong response in TLR4 pro-inflammatory cytokines. Interestingly, for some cytokines, you see that we've got some patients that they have, or some donors maybe, that they have responded like very strongly, while others not as much, while it's like a couple of donors that just not, that could be considered as non-responders. One disadvantage of the PCLS or for tissue culture in general is that it's actually you get very heterogeneous results or very high variation. But in the end, later on in the clinical studies, this is what you see. So I'm glad actually to see this kind of results, because in the end, it's a reflection of what we will see later on in a clinical study. And for the epithelial and endothelial injury markers, truth is that we were not able to replicate this finding at the same magnitude that we saw in the mice. And this is most probably attributed to some anatomical differences between the human and the mouse lung. However, we do see an upregulation of von Willebrand factor, which is another important endothelial injury marker. Sure, numerically it might not be, you know, like it's on the same order of magnitude, but maybe biologically it's still relevant. So on the same time, apart from ELISA, we also tried to measure this on immunofluorescence. And we were actually very happy to establish immunofluorescence stainings for PCLS in our labs, because we haven't done that before. And we have stained from for ZO1, this is zonusoccludin, this is a tight junction protein. Prosurfactant protein C is a common marker for alveolar epithelial cells, V-cadherin for endothelial cells, and DAPI for nuclear staining. And you can appreciate some differences. So from a quick view, someone could say that the red color is reduced and the green color, so possibly suggesting that levels of ZO1 and pro-SPC have been reduced, which is proportional that maybe this like epithelial cells or have been detached, or the tight junctions, we've got loss of tight junctions as well. Sure, for this you need a quantitative imaging approach, but this is actually something we are working on it currently with some help from bioinformaticians, and also to further quantify it and kind of answer the same question, like with additional experiments. We want to do some histology studies, mainly with H&E staining, so really to see what's happening on a cell-specific level. I mentioned like a image, like a 3D image quantification, because like, okay, for this one we have used the confocal microscopy. So with confocal microscopy, you can really scan through the tissue and take projections at the Z axis. So basically you have it's possible to create a 3D rendering of the entire tissue, or quite important piece of the tissue, so to get more quantitative information on that. And of course, we will collect supernatants and protein and extract the total protein from the slices to measure pro-SPC with Western blot. There's some activities ongoing for that. So to further characterize like the cell-specific responses of heme. Another important milestone is that we have actually managed to do bulk RNA sequencing. It took us quite some time to optimize our protocols. First of all, on even from freeze thawing and snap freezing, it's nice to see all the graphs here, but it's really a lot of work and long times, you know, spent in the lab to optimize these protocols. So this facility has been generated. These results were generated at the next generation sequencing facility of the University of Bern in Switzerland. And we got access to the raw data, and with the help of bioinformaticians, we were able to get a glimpse on differentially expressed genes. So here I'm showing to you the the volcano plot that you see, like in it's very common, like in bulk RNA sequencing, and we have set a p-value of 0.05 and a log2 fold change cutoff value at -+1. There is no rule of thumb for such you know, results interpretation. So since we prefer like to be a bit stringent with this, because it's the first time you know, that someone uses heme injury or aims to characterize heme injury on a complex tissue. And the good news is actually that there are some data sets available on lung endothelial cells. So at least there is the possibility to compare or deconvolute some signals with other publicly available data sets. Of course, it's not surprising that the most like significantly upregulated gene is heme oxygenase 1. So this is the enzyme actually that catalyzes heme in the liver. So yeah, we were happy to see that because also with other preliminary experiments with qPCR as well, we see that upon heme stimulation, we've got elevated levels of HO1. Another nice way to express bulk RNA sequencing result is to have this heat map, where here I'm showing to you like the top 20 differentially expressed genes, and we were happy to see that untreated or control, like in the yellowish bar here, versus heme treated, like the blue bar here, you see that there's a very good clustering. So really, these are the quite consistent, yeah, among different groups. So for the RNA sequencing, we have used 7. Lung slices from 7 biological replicates. With further discussions from the bioinformaticians, in this case, we're more on an in vitro case, you need at least 5 different repetitions. Maybe in this case, like an N of 3 wouldn't be enough. So you need like more samples, like or more biological replicates to kind of increase your statistical power. So yeah, we were happy that we would be able to work with 7 different biological replicates. And together with, alongside these two, the volcano plot and the heat map, we also did some gene set enrichment analysis. So what we see here on the x-axis is actually all the pathways that are involved. And on the x-axis, you see is the ratio of core enrichment versus the total genes inside. So these softwares basically have in the, we know from published literature and in the this public libraries or with the gene set enrichment analysis, the genes, so all the genes that are involved in specific pathways. So it's basically the number of genes that has been detected from each specific pathway divided by the total number of genes that is actually belongs to that pathway. So it gives us the involvement of the pathway and not, let's say, activation or inhibition of the pathway per se. This would be like a different algorithm to run. And so what we see here, of course, like the proteasome, which is very relevant for injury, and ER stress is also there. Also, it has been shown before published before in the literature in some in vivo settings, but not directly from heme injury, but in pathologies that heme was also involved. And we also see very strong pro-inflammatory pathways. So basically, it looks like that it's very, looks like it's very relevant for like LPS stimulation, or yes, I say like TLR4 stimulation, or microbial, or the same, or a very similar stimulation what you see from a microbial infection. So yeah, I hope like I have explained it to you, like this this graph. And also, if we want to talk about inhibition or activation of pathways, then you need access to other software. So, for example, in the past we have used Ingenuity pathway analysis from QIAGEN, and there also you can basically upload your data set and get some information on the activation on pathway activation. But I guess it's not a surprise to say that yes, all the pro-inflammatory pathways that are listed here will also be activated. So which is a strong prediction for activation of pro-inflammatory pathways. Maybe I move to the next slide. So to conclude, what we usually, what we have observed so far, maybe to start with the mouse PCLS, I would say that despite its limitations, it's more specifically, it's a very small tissue, so sometimes it's not so easy to generate many results, and maybe some techniques are very challenging to perform, like histology, for example. Still, it's a very useful model to establish optimal experimental settings for future experiments with more precious sample. And of course, we have to take into considerations all these anatomical and physiological differences also in cell types, and that not all the results could be translated into the human setting. And maybe another strong advantage of using mouse PCLS would be in the context of the 3R, so the reduce, replace, refine. So yeah, if you want to reduce the number of animal experimentation, or be more in accordance with ethics committee and applications, maybe it's a good alternative to in vivo testing, because also more cost effective, because you significantly reduce the number of animals. While on the human PCLS, we were very happy to see that we have established the transcriptomics procedure. So it's a streamlined procedure currently. And also on the same time, we're working with some confirmatory experiments more in the gene and protein expression. Also, it's we were very happy to see that we can mimic some key features of disease with underlying heme pathology. And now it would be important also to compare these findings with other published data sets, not only, I mentioned before the endothelial cells, but also with some other indications where we know that heme is plays an important role in the entire pathophysiology of the specific indication. So, and sometimes this can be tricky, because here we go with lung tissue, right? While the available data sets might be from signatures derived from blood, or bronchoalveolar lavage, or plasma, and for proteomics, for example, and so on and so forth. So it's really important to be very as accurate as possible on our interpretation, and also take into account the technical limitations. But yeah, I'm optimistic that quite a significant amount of work could be completed with this experimental setting. With this, would like to thank you very much for inviting me, and for all the nice session. And yes, I would be happy to answer your questions. I think we've got some time for a Q&A session. Yes. So yeah, yes. Thank you very much. And
wonderful.
Maybe I will stop sharing. And,
wonderful. Thank you so much. I just want to open up the floor now to all of our attendees. Please feel free to unmute yourselves and ask questions if you have any. And if you don't have any questions now, but think of ones later, let us know. Any comments or thoughts? Feel free to enter into the chat box too. And if not, we can wrap up, and we'd be happy to see you in the near future. All right, well, I just want to extend a final thank you to Dr. Fytianos. Somebody had asked, what is the thickness of your slices?
300 micrometers.
Okay.
So we started with 100, but in the end of it, they're so difficult to maintain, and it's actually recommended. So we wanted 100 because we're interested in microscopy and immunofluorescence, so we really wanted to scan through the entire tissue, but still, even if you manage to get 100 micrometer slices, it's not, you induce a lot of inflammation also from the blade. So if you work with 300 micrometers of thickness, then you get rid of some inflammation. So I would say it's a good, and this applies for human and mouse. So yeah, that's in my opinion, that's a good measure.
So you say that the thickness affects the viability of the tissue over time?
Viability as well and inflammation as well. So for example, even in your negative control, you might have quite a high threshold of inflammation.
Okay. Thank you.
Also, welcome. And also, yeah, for information, the diameter is 1 millimeter.
Wonderful. I have a quick question, Dr. Fytianos. So are there any tips and tricks you might have for increasing the viability of your PCLS?
Well, I would say one important thing would be, as we mentioned, the thickness would be one thing. And actually what we have done is to leave them in culture for 1 day. So basically, if we have it untreated and in the incubator for 24 hours, so to rest a bit, and then remove the medium and add fresh and replenish with a fresh medium after 24 hours, it has shown us that it reduces. It not reduces the viability, but we were happy with the viability level. So really, really high viability. I know there are other groups you can have them in culture for like 2 weeks or so, but for us, yeah, we actually don't work with like so long, treatments. So for us, I would say that it takes around a week or so, like from Monday to Friday, like to getting the tissue until have everything extracted and ready for downstream analysis.
I see. Got it. Very good. Well, if there are no more questions, I want to wrap up and wish everyone a good day. And again, say thank you to our speaker.
Thank you for having me. Thanks.
Thank you very much.
Goodbye. Bye-bye.
Bye-bye, everyone.